ਸਤਿ ਸ਼੍ਰੀ ਅਕਾਲ ਚਾਚਾ ਜੀ। ਵਧੀਆ ਜੀ ਵਧੀਆ।
ਸ਼ੁਰੂ ਹੋ ਗਈ?
ਹਾਂਜੀ।
ਚਲੋ ਚੱਲ ਠੀਕ ਹੈ। ਉਹ ਨਹੀਂ ਕੋਈ ਨਹੀਂ ਕੋਈ ਹੋਰ ਕੁਝ ਚਾਹੀਦਾ ਨਹੀਂ?
ਆਲ ਗੁੱਡ ਆਲ ਗੁੱਡ।
रोज इस टाइम में होंगी है क्लास के सवेरे भी होंगी है
ਇਸੇ ਟਾਈਮ
डेली शाम में मतलब 5 डे होंगी है
7 days
मतलब कि पूरे वीक जी शनिवार सोमवार को लेके एतवार तक पक्की ही हैं
10 दिन ਦੀ ਹੁੰਦੀ ਹੈ।
अच्छा 10 दिन लगातार एक क्लास आनी फिर ठीक है तो फिर हम फिर बुलाया हम कल कि नहीं
ਨਹੀਂ
हाँ
ਨਹੀਂ
और फिर अजय बाद च जरूर करी पेशी हो मतलब उत्थे का काम अज निबड़ गया ना फिर क्या तो चल रहा है सारे अंदर संसार से ही कुछ चलिया हुआ
ਗੇੜੇ ਵਿੱਚੋਂ ਨਿਕਲਣਾ ਇੱਕ ਵਾਰੀ।
गड्डी को ऊपर एक उड़ा लाती में लग रहा है
ਆ ਉੱਧਰ ਹੀ ਪਿੱਛੇ ਕਰਕੇ ਲਾਤੀ।
इधर लाती है शिपाल का दादे पूरे थोड़ा औखा लगता है
ਉਹ ਮੋਟਰਸਾਈਕਲ ਕਰਨਾ ਹੋਊਗਾ ਨਾ ਅੰਦਰ।
अच्छा समझ गई कि नहीं
ਘਰ ਜਾਣਾ
कल दिल्ली
ਕੰਮ
सवेरे सवेरे है ना वो थोड़ी सड़क खाली होती है ना चार दिन चलते कि ट्रैफिक सी
ਨਾ ਨਾ ਖਾਲੀ ਸੀ। ਆ ਟਰੱਕ ਹੁੰਦੇ ਨੇ।
हाँ ट्रक ज़्यादा होती है
ਟਰੱਕ ਸੀ ਵੈਸੇ ਠੀਕ ਸੀ।
थोड़ा जगह करके लॉकडाउन कोरोना करके थोड़ा रास्ता सड़का भी घट है 5 घंटे कुत्ते वाले आ गए 9-10 लोग
साढ़े 9 5 ਵਜੇ ਚੱਲੇ ਸੀਗੇ 7:30
ब्रेकफास्ट कितने करे
ਉੱਪਰ ਆ ਕੇ ਕੀਤਾ ਉੱਥੇ ਘਰ ਆ ਕੇ
हाँ
ਕੀਤਾ।
नाना जी के घरे
ਉਹ ਨਾਲ ਪੈਕ ਕਰ ਲਈ ਸੀਗੇ ਦਲੀਆ ਜੁਲੀਆ ਉਹ ਵੀ ਖਾ
ਸਵੇਰੇ
ਲਿਆ।
मम्मी ने बनाता हूँ उठ के जल्दी
ਉਹ ਹੋ ਗਏ ਸੀਗੇ। ਪਾਪਾ ਨੂੰ ਬੁਲਾ ਲੈਂਦੇ ਆ ਇੱਧਰ ਹੀ ਥੋੜੀ ਜਿਹੀ ਆ ਜਾਣਗੇ।
मैंने जी गंदा कर दिया मुझे डिस्टर्ब करता
ਨਹੀਂ ਨਹੀਂ
और के पापा कह रहे थे मम्मी कहने और
ਕੋਈ ਰੋਟੀ ਖਾ ਰਿਹਾ ਹਾਂ।
अच्छा रोटी ना दे हले
Hello, class. Very good evening, everyone. How are you all? Hope you are doing good at your end. A very warm welcome to the new session of Proteomics. Uh, so let more students to be added here. Then we can start our session. Let's wait for another 1 minute. So I think we have maximum students added here so far. I will start with the introduction today. First of all, a very warm welcome to the proteomics lectures to all of you who have joined us. Very thank you very much for to the Edufabrica who have provided this opportunity to teach you part of proteomics from my side. And thank you to the students who have joined this workshop to learn something, to intellect, to have some knowledge of related to proteomics. And then, yes, dear, it will be taken by the me. Uh, so, before we start, little bit of introduction of myself. Who am I? I'm a medical doctor professional. As per my studies, I did my BTech in biotechnology from Singh College in from PTU with 67.4 percentage. Then I did my master's with the University of Gießen, where my topic of master thesis was identification of novel and specific molecules involved in the regulation of tumor cell extravasation. Then I did my doctoral in 2017, Institute of Pharmacology and Toxicology in Göttingen in Germany again, where my topic was redox regulation of protein phosphatase 1 and E-stress regulation of connective tissue growth factor in cardiomyocytes. Then, after finishing my studies, I was also a research assistant in Germany while doing my PhD. I joined as assistant professor at Mata Victory College, where I was teaching following subjects, and afterwards I became as an online professor with Edufabrica, where I'm teaching various workshops like molecular biochemistry techniques, biochemistry, principle of farm biotechnology, principle of genetics, and this is the recent added topic, proteomics also. So I'm creating online video lessons for all of you, and one of my publications from my PhD work has been published as a work with the reduced activity of PP1 alpha under redox stress conditions and consequence of GSH mediated transit formations in scientific reports. So that was a little bit about me. Uh, Tell me about you, students. So, yeah. Let me check the disturbance. Mic testing. Hello students, is the voice is okay now? Is it better? Are we on the loud voices okay now? I have fixed it a bit. Uh, is things are better now? Okay, that's good. Beautiful. Awesome. I will keep this voice in this manner. So background disturbance. Okay, I will start with some. I have given my introduction itself so far. Now it's time for us uh, to tell you about how we will phrase our internship and assignment things. So there's two things. One is students who have opted for internship, they have to do one review article in which you have to write one topic of your own choice related to proteomics, and you can write one review article out of it. So that would be one task, and still some of the work that of the assignment work that I'm gonna suggest you is that um, that whatever you learn from one thing from today onwards, you have to maintain in an assignment way a summary of each day. You have to write a summary of each day in 2 pages and send it to me on my email ID. So my email ID, I'm sharing it here. I will also share in the WhatsApp group. So one thing is assignment, and this is not compulsory to send me the assignments, but you can, if you can send it, it will be really great for me. That will make things easier for us and for you also, so that you can have some summary of what you have done so far that you can write. So from today onwards, you can start writing review, and afterwards 5 days more you can get it to write the review. So almost 15 days. It's more than enough. Just I will share with you now how should be the review article should be look like, and how should be the review format of it. So review format sounds like this. So there should be a title page, title, your name, date, abstract. It should contain almost 200 to 300 words. Includes a summary of the review question, primary study reviewed, conclusion of the study. An introduction, write the topic of the study which serve as identification of sentences. It should indicate what articles contains. And further, the body should include the subtopics of addressing the topic. And conclusion, it should briefly state your rationale of your review, the purpose of the article. Then at the end, literature cited. Use a standardized reference system or using an MLA system. So in this way, you have to share your review article. So I will share this part with you. Just wait a second. So students, This will be the review format that you have to choose. I can share some examples with you from the previous experiments. So something like previous students who have done very nice from my MBBT workshop. So something like abstract is there, name of the topic, people, the topics of the the from the university they belong to, abstract. Then there's abstract in a very short and nutshell, keywords, introduction, then body of your review, then all the points are from the previous research what has been done, then what is the present is going on, what is the future is about that. So keeping the all time scales in upfront, then comes at the end references of the same thing. So this is how you have to maintain your your review articles. So I will share these with also you. Just wait. So these articles that I have so far, all these reviews has done by the student itself. So we have just provided them with the format, and they can choose the topics of their own, and voila. So they also, some of the students have published them in a good journal. So whenever you are writing a review, so please take care that you write with related to proteomics topic. So nothing beyond proteomics, because in your certificate, it will be written that while doing the proteomics workshop, you have done this review article. So keep that thing in mind while doing so. So I will share one more article. And about assignments, so this is one example of assignment that somebody did. Each day he was writing, I don't know, he or she was writing about it, about 2 pages in a very nutshell. But this was very nutshell actually. It's not that much short. Sorry to say that. Maybe this one is good. Yeah, this one is good. So something, these are the assignments that students were sending me. This is one example of the same. There you go. Hello, hello students. Yeah, I will. You can also, some of the students have published their articles for publishing also. They went for that. So it's just find some free journals where you can publish, and things could work positively for you. So this is also possible. It's no problem in that. Okay. So I have shared 3 things with you on the WhatsApp group, mainly the review article, review format, mesoporous, and these 3 different things. So you students, you want to be on Google Meet, okay? But students, what happened at the Google Meet? I have tried many workshops, so there's option of adding students automatically. So that's where I feel really problematic. Um, So each time, whenever I'm giving presentations, some students just pop in, and they want to be part of the workshop, and I cannot stop that. So I have to stop in between and come back, stop in between and come back, stop in between, come back. So this goes forever. So that's why I opt for uh, this thing. So today I had Zoom. Zoom is it's both actually. In Zoom, it's very less students. I guess it's not 100 students allowed. Maybe 50 students free are allowed. Okay, I will provide you few generals soon on the WhatsApp itself. So don't worry about it. Um, so this is so far is the best platform to go for. Today I'm actually not at the home, but if I'm at the home, so the problems that we are having with the sound and things might be not occurring again. But I will take care that this is not happening anymore. So don't worry much about it. Don't worry. Okay, so we are done with the basics. Uh, about everything. If and any of you, if you are looking for some masters or PhD in abroad, so you can ask me anytime. So I can, if I have free time, I can make sure I can. But okay, I will share the review topics for this also. Okay. So this was some introduction part for the starting the workshop. So hope things are clear, better on the undertaking. So let's start the workshop today. So today is the introductory class. So where we'll be dealing with the introduction to proteomics and protein stability, or we just stick to introduction to proteomics today because today is not such a I'm not in a good position to teach you all in a good way. So let's fix the next lecture to tomorrow so that we can have better things available for you. So today I will take only one lecture, that is introduction to proteomics, and the rest we will do tomorrow for today what we have. So students, in this lecture, you will be acquainted with the basics of proteomics with special emphasis on the proteomics techniques to elucidate the physiochemical properties of proteins in general. Then they will be getting depth knowledge of how thermodynamics help in proper folding of amino acids chain in 3 dimension to form an active biomolecule. This course will strengthen your basic concept and pave the ground for better understanding of other related subjects taught in the biotechnology. So this outline of the lecture is divided into these parts. What is proteomics? Why study proteins? How to study proteins? Discuss proteomics tools and methods. So what is proteomics? It's the analysis of complete complement of proteins, identification and quantification, modification, interaction, and activities. So deal with all these parts and how these changes are happening in biological response. So if it's the Department of Genetics and Biochemistry, they are just searching for things, but the Department of Genomics and Proteomics department, what they will do, they will get all the things that they were looking for, like all the proteins, mic protein, P53 protein, RB protein, ATM protein, Notch protein, UB protein. So all are being extracted. This is the one cartoon showing you that. So proteomics is the analysis of protein complement to the genome, and genomics uh, and protein is for proteomics. So from one organism will have radically different protein expression in different part of its body, in different stages of its life cycle, and in different environmental conditions. So each body, each part have its different life cycle in different environmental conditions, and they will behave differently accordingly for the same. So from gene, transcript will happen. From transcript, the protein is happening, and the study of gene is called genomics, and study of protein is called proteomics. And the large-scale study of proteins, while it is often viewed as the next step, proteomics is much more complicated than the genomics. So proteomics, what we are studying so far, is much, much more complicated than we thought of the genomics part. So in here we can see different cell cycle of butterfly and also of the carnage stages of human beings, human development. students, I have to check in between with your. So I will share all the PPTs with you after the slides are done. Uh, so because I was having a class of ambiguity before, so it was getting off automatically. So I might need to come back and forth to check that everything is going fine or not. So I cannot see things like that. So here we can see that radically different protein expression in different part of its body in different stages of life cycle in different environmental conditions. So egg will have different proteomics than caterpillar, and and sorry student, sorry. Yes, me sir. अभी तो ठीक चल रहा है सर. ओ, ठीक है सर. ओके सर. So students, uh, I mean, proteomics is something not hard. It's one of the branch of science where it is quite needed, because whenever we are studying about proteomics, we are studying more about I did my PhD in proteomics itself. My whole I will share one of my PPTs with you what I have done during my time for proteomics. That it was more about interaction with the mass spectrometry, some analytical techniques, a lot of Western blotting techniques. So all my PhD was based on the proteomics. So if you're working in the wet lab, most of your work will be done with the help of uh, in the field of proteomics pertaining actually. So whether it's a Western blot technique, mass spectrometry, or analysis of the post-translational modifications, all these things are coming up into the proteomics. It's not hard, but I will try my best to teach you all the techniques that are related to the proteomics, so that at the end you can think what is the best for me, whether I should choose this or not. But most likely it's one of the hot topic actually. Proteomics is quite hot, and everyone wants to be have one proteomic specialist in the department, and then the group studies. So let's continue with the topic. The genome is a rather constant entity, the proteome different from cell to cell, and is constantly changing through its biochemical interaction with the genome and environment. So they are changing as environmental is changing, as the genome is changing. So they are constantly having a different scope of life. The last case study of protein, while it is often viewed as next step, proteomics is much more complicated than genomics. So as we see the genomics, mostly scientists they're looking for at the gene level, but even in the proteomics level, it's even more complicated. But once you solve this proteomics part, you can solve many unsolved questions. So like expression, 3D structure, interaction. So all these things are related to the proteomics pertaining part, and they are somehow correlated by bioinformatics also. So if you see the expression as a 2D gel, you can see the proteomics results, a 3D structure of results with the help of bioinformatics, and all these things are possible with the help of interaction between each other of these things. So proteomics is basically multidisciplinary, which has bioinformatics, biology, protein biochemistry, analytical chemistry, molecular biology. So proteomics research, basic research is to understand the molecular mechanisms underlying the life, applied research, clinical testing for protein associated with pathological states. So proteomics itself has subcategories, as we have discussed, such as proteome mining, functional proteomics, structural proteomics, protein-protein interaction. protein translational modification, protein expression profiling. So within them, within the protein mining, you can go for drug discovery, target ID, differential display. Whereas the functional proteomics, you could go for yeast genomics, affinity, purified protein complexes, mouse knockouts. So with respect to that, you can, with the help of proteomics, you can hint so many things in such a wide manner. For example, hemoglobin picks up oxygen in the lungs, travels through the blood, and delivers it to the cells. So here, oxygen, you can see that it is made up of your hemoglobin is made up of Hb beta, Hba, and Hb beta. And then hemoglobin is making two RBCs here. You can see this. So sickle cell disease is caused by single amino acid chains. So normally hemoglobin beta, it has mutated HBB beta, such as you can see GAG. GAG is like here is epinephrine, and this GTG is converted to GTG. So you can see that A is mutated to T into valine. Phenylalanine is converted to valine, and it leads to the mutation in of the sickle cell disease. So, in summary, what is proteomics? It involves the study of proteins. Proteomics is a multidisciplinary, and proteomics is being applied to both basics and clinical research. Okay. Yes, बच्चे. So today is a short lecture for today, but I will compensate it tomorrow. So don't worry about it, because I have some issues at the moment, so I cannot go for longer. I'm in somebody's house, so I have to stop it early. Yes, space biology, marine biology, geologist. So everyone is actually looking at the protein. At the end, we are all made up of genes. I agree. We are made up of DNA. Then RNA is made. Then from RNA is protein is made. So at the end, we are all at the end. Protein is made by the help of transcription. So because of that, our results that we are looking for, if we see at the protein level, things will be much better and much cooler. So why to study proteins? Because it has motion, actin, contract muscles, enzymes like protease, degrade proteins, regulation of insulin, control blood glucose, defense antibody, fight viruses, sport keratin, forms hair and nails, and transport hemoglobin carries oxygen. So all these parts are done because of the proteins. So proteins are the large complexes molecules that serve diverse functional and structural roles within the cells. So proteins are composed of amino acid blocks. So here there is a variable. Then there is a carboxylic acid. Then there is a amino acid base. So amino acid 1, amino acid 2, together they make this peptide bond. H2O is removed, and there is a peptide bond is made just like this. Dipeptide bond. So these are various non-polar, polar, hydrophilic, acidic, basics amino acids. Here you can see various molecules as per the structure and the molecular chemical properties they have. And proteins are the chain of amino acids. So here you can see various amino acids. They bring it together and work like this. So short chain of amino acids are called peptides. Proteins are polypeptide molecules that contain many peptide subunits. So translation is the synthesis of proteins in the cells. Then there is a large subunit, small subunit come as a ribosome. Then a methine transfer RNA comes and starts the translation. Codon 1 methionine AUG is the one from where this starts. Then comes your alanine as a transfer RNA. Then comes codon 2 GCC, that is alanine. And then comes the next TRP, tryptophan. It's got attached. So this is how your translation will keep going on. Your ribosome will move forward, methionine, alanine, and tryptophan. So your empty tRNA will remove. Codon 4 will come like this, codon 5, and at the end your protein is made, methionine, alanine, and like this you have your whole new structures. So any questions so far, students? So I hope this basics was clear. So basically, if you see the applications, there are wide variety of applications I have discussed as of now, like this one. So proteomics technology is having, so here you can see conventional techniques. Just wait a second. So proteomics technologies could be conventional techniques, advanced techniques, quantitative techniques, high throughput technique, bioinformatic analysis. So conventional technique includes chromatographic based technique, ELISA, Western blotting. So within these techniques, you can have ion exchange chromatography within the chromatography, size exclusion, affinity chromatography. And within the advanced technique, you can have protein microarray such as analytical protein microarray, functional protein microarray, reverse phase protein microarray. And further in the advanced technique, you can have gel-based mass spectrometry, advanced sequencing. In gel-based, it could be SDS page, 2D page, or 2D dig. It's a new kind of uh, electrophoresis. Then there's a quantitative techniques like ICAT, SILAC, ITRAC, XRIC. These are three different quantitative way of doing so. So we will discuss about each of them: X-ray crystallography, NMR spectroscopy, sorry, bioinformatic analysis. So these are the various applications which have been carried out whenever you will go for research. Okay, I will just because I will forget later, so I will share it now. So I shared the same paper. So is done. So, okay, we discuss about proteins also. Before we do, let's have our daily routine. First, we finish this workshop. Then maybe tomorrow I can show you further about that with a virtual lab maybe. So proteins have specific architecture. Here you can see there's four heme groups are present of the hemoglobin like this. They have specific structure. So somebody was asking about me primary, secondary, and tertiary. So this is how sequence of amino acids is there in the primary structure. Local folding maintained by the short distance interaction. Then additional folding maintained by the more distance interaction. In this case, tertiary structure. Then they are then these structures are joined together and make a quaternary structure by interchain interaction. So this is proteins arrive at the final structure in the ordered fashion. So what we studied here so far, we have studied that biological workhorses that carry out the most of the functions within the cells. So several diverse functional and structural roles composed of amino acids that are covalently linked by peptide bonds. We have studied that synthesize during the translation process and must fold correctly to perform their functions. So, so basically, what happened is, uh, yes, CRISPR is actually RNA biology, which we have mutation of one part. So it's more about RNA biology, I would say. And about assembly of protein, first is your primary structure, then comes your secondary structure, then comes the third tertiary structure in which you have beta plates, alpha helix structures, then comes the quaternary structure. This is how things are made. So I will make sure I will write it down to have a proper video for the same. If somebody want to be more interested with the protein assembly, I will make sure to have it. So, any more questions? Okay, so far so good. You can, बच्चे go for PubMed. You can look for many papers, lot of papers in PubMed and CBI. So internship project is बच्चे is the same that what we have taught. I have told you is about the review writing. I have discussed in the earlier class. So I have shared a lot of review articles also with you. Just go with that. Yeah, research is also good option. Yeah, that's true. But try to find out. You can find some of the abstracts you can read from them. Otherwise, we need from the university website, from the university console that we can have it all for free. So just check it for that. Plus nature, yes, yes. Mentor. So yeah, we will help you. Just write down your papers. Me and I have a colleague. We will both be there for you for publication also if possible, and we try to get it done within time. But you have to work on your own also. So just make some group within your friend circle, and try to go for it. Yeah. No, no. If you have not bought the training program, then you don't have to buy. You have to do this review thing. Only if possible, just try for Sci-Hub for full access. Yes, that's also a good option. Go for that also. Right, right. So students, I have limited time today because of the outer space. Uh, okay, I will find some articles for you for tomorrow and will let you know. Don't worry. This will be for 10 days so far in the proteomics. So don't worry, don't worry. So proteomics tool to study proteins. So first you have the protein isolation, protein separation, and protein identification. In the protein isolation, you have mechanical methods like grinding, breaking up open the cell, centrifugation, remove insoluble debris. Then chemical methods is like detergent, breaking up open cell compartments, reducing agents, break specific protein bonds, heat, break peptide bonds to linearize proteins. So all these things are being taken care. So protein isolation procedures. So first you find a sample, pick it up, grind sample in buffer, then transfer to the tube, then heat the sample, centrifuge to remove the insoluble material, then recover the supernatant, and keep solution for gen analysis. Pure protein solution is there in your Appendorf. So protein can be isolated by a variety of samples. Proteomics includes the use of both uh, mechanical and chemical methods to isolate proteins. Opening a cell or cellular compartments, breaking bonds, and linearizing proteins, removal cell debris. So pure protein solution, isolated protein X, you can remove it like that. So afterwards, protein separation SDS page is done. First is pure protein solution, tube 1 and tube 2. Tube 1 includes increased complexity, decreased protein ID. And the second case, decreased complexity but increased protein ID. How to separate proteins? Separated intact proteins is to take advantage of the diversity in the physical properties, especially the isoelectric point and molecular weight. So isoelectric point, and based on the molecular weight, you can separate your proteins. So for that, there are two methods of separation: SDS page and isoelectric focusing. SDS page full form is sodium dodecyl sulfate polyacrylamide gel electrophoresis, and isoelectric focusing IEF. So SDS page, sodium SDS polyacrylamide gel electrophoresis is a widely used technique to separate proteins in solution. Molecular weight is a mass one molecule. Dalton is a small unit of mass used to express atomic and molecular masses. So page is widely used in proteomics, biochemistry, forensics, genetics, molecular biology. A major component of polyacrylamide gel is uh, bisacrylamide, TMAT, ammonium persulfate, free radical initiator. So all these things are required to in the polyacrylamide gel. So what is the structure of these structures? Of acrylamide structure look like this, bisacrylamide look like this, and ammonium persulfate is SO4, free radical initiator. So in polymerization, what is happening is bisacrylamide cross-links CONA2H are attached like this, and making a polyacrylamide like that. So sodium dodecyl sulfate SDS, anionic detergent SDS unfolds or denatures proteins. Before SDS, we have like this, H like this, hydrophobic and charged H groups, and after SDS is like this, minus minus minus. So initially your protein is in the uniform, is ununiform, but at the end it's become uniform linear shape and uniform charges to mass ratio after SDS. Before SDS, it was just charged R group and hydrophobic areas were present. So in one-dimensional polyacrylamide gel electrophoresis, so you have a negative and positive anode, standard sample 1 and sample 2, and you have bromophilic blue dye front like this. So you have, during the SDS space, separates according to their molecular weight. So there's a standard sample 1 and sample 2, and you have your proteins to be separated like this. So these are your various protein bands, big bands present like this. So there's a protein X with 25 kilodalton here, and we have separated this. So this is also image of real SDS page, looks like this. So from 250 kilodalton to 20 kilodalton goes like that. So afterwards, you can do two-dimensional gel electrophoresis. That is, first dimension isoelectrofocusing, then second dimension is the SDS page. It's most widely used protein separation technique in proteomics, capable of solving thousands of proteins from the complex samples. Isoelectric focusing is the separation of proteins according to the native charge. So in isoelectric focusing, you pH at which net zero is, the charge is zero. At that point, your proteins start to separate from each other. So here it's like isoelectric point of a protein is here around 8. So like this, you can see a protein sample has been arranged here, and protein moved from negative to positive with a pH gradient from 3 to 10. And then you have variety of proteins being present on this sheet with neutral at pH 3, and first dimension isoelectric focusing to be like that. Second dimension SDS page, uh, so second dimension SDS page, afterwards the first one is done, it looks like this. So this is your isoelectric focusing point. This is 2D gel for different pH, and these are separated of their respective size. So this is a results from the developing leaf, Arabidopsis developing leaf. So I hope I am still online, it's not disrupted. Okay, all is good, it's going fine. So in this case, we have earlier find out previously 25 kilodalton with the normal gel, but in the when we run the 2D gel, we can also find out 25 kilodalton component like this here. So this was 25 kilodalton base pair protein X at the pI 5, at the pH is 5. Uh, Isoelectric focusing point is 5, then it's around 25 kilodalton is the same protein here. So you can identify various proteins like this. You can check in the forensic science also to check samples taking from the forensic place where some crime scene has been done, and also from the non-crime scene done. So you can check between both how things are being prevailed. So first DGE versus 2GE. High reproducibility, modest reproducibility, quick and easy, and this is slow demanding, where this is separately solely based on the size, and this is separate based on the pI and size. This is modest resolution dependent upon the complexity of the molecules, and this is the high resolution not dependent upon the complexity of the sample. So in summary, what we have discussed here is protein separations takes advantages of physical properties such as isoelectric point and molecular weight. SDS page is a widely used technique to separate proteins, and 1DG is a quick and easy method to separate proteins by size only, and 2DG combines isoelectric focusing and SDS page to separate proteins by pI and size. So this was about separation of proteins by various techniques we have discussed. Now comes the mass spectrometry. In this mass spectrometry, this was, so you can see that you have a protein marker, and purified target protein is, you got it, and then you eluted this trypsinized with protein band. So you put some trypsin inside that, and then also add CH multi-matrix to it. So you have protein and matrix both, and then you add your protein and matrix mixed together spot on the spots of the multi-target plates. And these multi-target plates, you put some laser beams from the top, and the time of flight tube is done, and then you see these peaks. Then the ion detection is done at this point. Then with the help of MSMS database searching, you find the peptides, that what you're looking for. So peptide mass fingerprinting, how you do it? So first you have intact protein, you digest that protein with the mass spectrometry, then you have different masses, and then you have protein ID like that. So make proteolytic peptide fragments, digest the protein into peptides, measures peptide masses, weigh the peptides in a mass spectrometer. Match peptide masses to the protein or nucleotide sequence database, compare the data to known proteins and look for a match afterwards. We use the enzyme trypsin to digest proteins into peptides. Trypsin cuts into after lysine and arginine. So like this, you have protein X, and with the help of trypsin, you cut it. So like this, it cut from here, and like from here, we cut it again. So we have three different polypeptides with the K and R lysine and arginine being attached there. So basics of mass spectrometry is how does mass spectrum identify unknown proteins? It determines the mass to charge ratio. The mass spectrometer very accurately weighing the scales, third or fourth of decimal place. And then we weight these peptides with the help of mass spectrometer. So their sizes after the mass spectrometer came out to be 1106.55 Dalton, 692.31 Dalton, 1002.37 Dalton. And mass of peptide should be compared with the theoretical masses of known peptides. So these masses are known to be present in our data, informational data, and like this. So we get milk, like this. The different things are there, different peptides are there with the different sizes. So we can compare these peptides with this size, and we can say like 1106 to be like we get milk like this one. 692.31, 692 is, it would be A tier. This would be this peptide, it would be 11001002, it would be this one. 3 5 3 7, it's a 3 7. Anyhow, so we have found, yeah, this one, many QWS. So mass of peptide matched to theoretical masses known peptides using a computer program. So unknown peptides have been identified. We get milk, S tier, many cows. So these are the various peptides of these unknown peptides being cutted out with the help of trypsin, and we can let us know that these peaks belongs to these kind of peptides. So we get milk after many cows. This is one just strange example. It doesn't have any meaning. So summary, proteins are digested into peptides. Peptides are analyzed with the mass spectrometer. Match observed peptides mass to the theoretical masses of all peptides in database. And assemble those peptides matches into proteins identification. So in this topic, you have studied thoroughly about SDS page and 2D gel electrophoresis, basic principle of mass spectrometry as a tool in proteomics. At the end of course, you should learn the practical concept of how to characterize unknown proteins through proteomics tools actually. So these are the various references. Thank you very much for today, for being there. But a multi-matrix is the matrix is like a what shall I say? Like kaddukas hota hai na, aapko kya kaddukas in English is yeah, it's a kaddukas actually, same like kaddukas. This one. So here you put your samples on these. So here the laser light, they came and they put it and ionize your samples and to check the protein, which peptide it is, which peptides are present in that protein. So this is how like A, B, C, D, G, H, and this is 1, 2, 3, 4, 5, 6, 7, and 12. So this is 96 well plate matrix, multi matrix. Okay, I will share the PPT in the group now with all of you. So please ask your questions not in the WhatsApp group, but on the YouTube. It will be better. So yeah, one important part of today's lecture is it will be recorded, and you can watch it anytime with the link that I've shared on the YouTube. So you can watch it anytime you want. So I already shared the PPTs with you. Just have a look there. References, books. Okay. So you have the PPT. You can watch it also. Principle of Proteomics, Introduction to Proteomics, Protein Engineering and Design, Mass Spectrometry textbook. Also by NPTEL, NCBI, National Digital Library. So all are being taken care while doing so. So I will repeat assignments. I feel funny. What about assignments and all? So by tomorrow I will be home in Delhi. So I will share with you all the topics and make things done for you. So don't worry about that. So everything will be done as per things are. I will share the review topics also. No worries. Okay, students. If you have no further questions, I have to go now. Maldi, we will discuss Maldi MS and ESI MS. We will do it in more time. So don't worry about that. So sorry if I have a 1 hour class today, but tomorrow I will try to make it more because I'm in somebody's house and we are allowed to work till 9 today. So I will make sure that things will be done more for tomorrow. Today was an introductory class so that you can have a something simple to understand, nothing so difficult, and we will go for more classes like this. Timing will be same from 8 o'clock, from 8 to 9:30 or so. So I will try. Yeah, assignment is not compulsory, but if you do it, it's good for you and for us also uh, to have a everyday check that what we are doing. Session before 8. I mean, I would be happy to have session in the morning. Uh, something like 8 in the morning, but students will not agree, I guess. So students will not agree. Yeah, mass spectrometry we will discuss in details. Don't worry. So this was just basics about it, about everything actually. So if we have no other further questions, thank you very much. Call it Lex. Okay, lot lectures in the morning. Let it be evening. Everyone has paid for the evening classes, so we can't change. So evening would be from 8. From 8. It has to be from 8 to 9:30 or so. Mainly, our task is to complete each topic, what we are going through, and clear these things for us. So that's the basic part. Yeah, evening time sounds good for everyone. I have some classes students from 5 to 7:30 first, then 8 to this one is, so I cannot adjust. That's problem. Okay, anyhow we keep it the same timings so far. So don't worry, proteomics is been used in all the field of science and every part of the field. So all the basic scientists they need at the end proteomic specialist for studies. So okay, good night, students. It was pleasure to see have you to have my students, and I will see you tomorrow at 8 or 7:45 with more interesting topics, more things to be shared with you. Thank you very much. Wish you all good night. Stay safe, keep smiling, and be awesome. Yeah, that's the key. Be awesome. Okay, good night. Bye.